Everyone, today I would like to introduce to you an article we recently published in MITF on the duck pan-genome. Titled Duck Pan-genome Reveals 2 Transposon Insertions Cause Bodyweight Enlarging and White Plumage Phenotype Formation During Evolution. Recent studies have found that many phenotypic variations are caused by structural variations. And further discoveries show that many structural variations do not exhibit linkage or have low linkage effects with nearby SNPs, indicating that many phenotypic variations caused by SVs cannot be detected by SNP-based studies. However, current genomic analysis mainly relies on a single reference genome, which cannot represent all the genomic information of the species, greatly affecting the identification and analysis of SVs. In recent years, pan-genome studies have become more common. A pan-genome can integrate the genomic information of multiple varieties of individuals, representing all the genomic information of the species. The current forms of pan-genome presentation are mainly divided into linear pan-genomes and graphical pan-genomes. This study still adopts the form of a de novo linear pan-genome collecting 5 publicly published chromosome-level genomes, optimizing and combining the PSV-CP and PPSP-CP processes and newly identifying about 40.98 megabits of sequences not found in the reference genome, which compared to iterative assembly processes can return a large number of new insertions to the reference genome. In addition, we used high-depth second-generation sequencing data for SV calling, identified over 100,000 SVs and constructed evolutionary trees and population structures based on SV data, which can effectively separate wild ducks, domestic ducks, and commercial pecan ducks and is consistent with SNP clustering evidence. To identify structural variations that potentially affect the domestication and improvement process, we used FST and Fisher's exact test, identifying 999 potentially selected SVs during domestication. Functional cluster analysis found that they might be related to neural system development, bacterial response, and morphogenesis. Similarly, we also identified 953 potential SVs during the improvement process, which might be related to reproduction and spermatogenesis. Notably, during the improvement process, 2 interesting SVs were found near the IGF2BP1 and MITF genes. Analysis found that 24.37% of the newly identified sequences were transposon sequences, while only 9.49% of the whole genome level was identified as transposons, indicating that many new sequences are mainly due to transposon insertions. More and more evidence shows that transposons have a significant impact on the production of SVs. To verify this finding, we conducted a chi-square test and found that the frequency of SV occurrence is significantly related to the frequency of transposons. To verify this conclusion, we further analyzed the TE located upstream of IGF2BP1, del 00154411, and the TE inside MITF, del 00130156, for the TE upstream of IGF2BP1. Further natural population evidence proves that this variation is significantly related to body weight traits and has been strongly selected during the improvement process. The transposon insertion in the intron of MITF, we found in the verification population that the transposon insertion is significantly related to the white plumage trait. Through race experiments, it was found that the transposon insertion produced a new chimeric transcript, significantly reducing the expression of the original transcript. We also conducted an evolutionary analysis of Gypsy transposons, finding that duck Gypsy transposons have undergone significant expansion compared to chickens and formed 3 branches. The Gypsy transposon on IGF2BP1 and the transposon insertion in the intron of MITF are quite ancient, occurring 3.88 million years and 0.6 million years ago, respectively. In summary, we present the first duck pan-genome capturing 40.98 megabits new sequences. A substantial number of these SVs were associated with traits related to domestication and improvement. The Gypsy insertion located at the promoter of IGF2BP1, to our knowledge, explains the largest effect on body weight among avian species.